Okay, maybe we could start by looking through the the discussion prompts. Well, maybe we've got a relatively small group. Should we start by just everybody giving introductions? So I'm David Osumi-Sutherland. I work at the EBI. Actually build ontologies for a living. I'm a developmental biologist by background. Want to go.
So whoever's showing their screen, can you expand that, because for some of us the type's coming up really small.
Okay, I'll make it.
Thank you.
Is that better? Sorry, it's being on a large screen. There we go.
Awesome, thank you so much.
Do you all want to introduce yourselves?
Hi, Deborah Hoshizaki. I'm NIDDK. I'm a project officer working both with HubMap and with the Kidney Precision Medicine Project, which is an NIDDK sponsored project. I lead the Data Visualization Center. I'm also the program manager for Rebuild a Kidney and for GoodMap, which is a molecular atlas of the developing mouse and human kidney and lower urinary tract. I will do my best to kind of lurk in the background, but knowing me, I'll probably be popping in with comments.
That's a good segue. Do you want everyone to introduce, or only one?
Yes, please. Yes, please. I think it's.
So hi, my name is Becky Steck. I'm from the University of Michigan. I'm going to go after Deborah, since she and I work together on the Kidney Precision Medicine Project, where I lead the software development team that's responsible for our Data Visualization Center, which is where we're doing all of the visualization, storage, collection, analysis of the data we're collecting. I think I'm going to be a lurker here, but we have been doing a lot of work right now in developing, actually improving the cell ontology and Uberon. So I may pop on video wise if I have things to help with.
I can follow on if you don't mind. My name is Todd Valerius. I'm part of GoodMap and Rebuilding a Kidney. So I work with, and KPMP to some degree. So I work with Deborah and Becky as well. I'm at the Brigham. I serve as the biocurator for GoodMap and RBK. I'm also a long-suffering developmental biologist. And so that's where my fascination is. And we have been thinking a lot about anatomical ontologies like Uberon and down to the level of the cell as well, and trying to get this all to link up so that we can also use our single cell and single nuclei data in this way.
Let me take a little bit. Marion, do you want to go next? Sorry.
Sure. My name's Marion. I work at the EBI in Cambridge. I work in the HCA DCP team and work with contributors that want to contribute their data to HCA DCP. So help them with ontologies and metadata, and trying to also, we work a lot to improve some of the ontologies. Yeah, that's me.
Ed, do you want to?
Sure. Hi, my name's Ed Lean. I'm an investigator at the Allen Institute for Brain Science. I'm also involved in the Brain Initiative Cell Census Network, and interested in this topic because we're all creating these very detailed data-driven cell classifications, and it's time to build some real robust ontologies around them.
David Rogers, do you want to introduce yourself?
Sure. Yeah, my name is Dave Rogers. I'm working on the HCA data portal and the data browser. So we're consumers of these, trying to navigate and present them and allow search over them. Very interested in this topic. Yeah.
Ambrose?
Hi, this is Ambrose Carr. I'm a product manager at the Chan-Zuckerberg Initiative. We're a contributor to the data coordination platform of the HCA, and we also build the cell by gene application, which has some capabilities to allow user-curated annotations. So very interested in this topic.
Sorry. I hope did I pronounce your name correctly? Idan?
Idan.
Idan.
I'm building a data wrangling team at the ENCODE Data Coordination Center in Stanford.
Nikolai?
Yeah, I work at Northwestern on the lung seed atlas, and we're publishing our data sets too, and classifying cells. That's all very needed.
Bonita?
Hi, I'm Bonita, and I am a data wrangler at the ENCODE Data Coordination Center at Stanford.
Alexander?
Hi, my name is Alexander Misharin. I work with Martin Nawin. I work with Nikolai Markov. We're part of lung biological network. I'm more on the biology side, generate samples and annotate samples. Looking forward to actively contribute to cell ontologies.
And Hansheim? Did I miss anybody? Okay.
This is Jonathan Silverstein, and I'm one of the PIs on the infrastructure within HubMap, and I'm a recovering general surgeon and professor in biomedical informatics at the University of Pittsburgh now, and very heavy ontology user of many, many kinds, clinical, molecular, and biological, and integrating them for use across HubMap. So we badly need something at the cell level that isn't yet there as far as I can tell, and so I'm very interested in where this goes. So I intend to lurk and listen to where y'all think it needs to go, because I operate at the much higher level in general than the cells, and it's working my way down that far. So.
Have I missed anybody else? I think we're about...
Yeah, hi, I'm Martin Nawijn. I'm from University of Groningen in the Netherlands, and together with Sasha Misharin, I'm one of the coordinators for the Lung Biological Network. And we've worked a lot on integrating the data set for the COVID-19 analysis. So actually trying to harmonize the ontologies, and doing so, we really realized how difficult this is. So I'm hoping to learn as well as contribute to this session here.
Okay, I think we need a couple of volunteers to be scribes. I will try to take notes as we go, but I may be talking as well. So any volunteers? Otherwise, I guess I might have to nominate people. No volunteers. Okay. Let's try and keep it gender balanced, shall we?
I volunteered. This is Becky Steck. I can help.
Becky Steck. And then Dave Rogers, would you be able to take some notes as well?
It'd be difficult for me. I'm juggling.
Juggling kids. Okay. Yeah. Yes. I might be later as well if I. Yeah. Okay. Any other volunteers on this? I know here. Ambrose, do you want to take some notes?
I think it could be hard for me at the moment.
Okay.
Sorry.
All right. Well, okay. Well, I'll try to take notes as well with Becky and anybody else who wants to jump in and just add notes. So I think what we'll do is put rough notes in the green section, and then we can move into the blue section to try to record, certainly probably in session 2, to try to record what we're actually gonna try to do. But let's just keep in session 1. Let's keep notes in the first green section here. Okay. So where should we start? So the discussion prompts we started with here. So major uses. Maybe if we go through these sort of major use cases of why we want, what we want to achieve by using cell ontologies, and then see if there's anything that's obviously missing. I'm sure there will be. So major use case queries for cell ontologies and data annotated with them. One I would say is maybe having these things link up to anatomical locations in some way. Another, having them link up to sort of key markers, maybe for example, clinical markers, or markers that are used classically. But then there's the question of how deep we should go in recording markers, because obviously there's a tricky line here to navigate between data and metadata. You know, to what extent should we have terms that annotate sets of markers versus including those in the ontologies? Being able to query by gross cell types at the very least, you might want to query across multiple data sets for leukocytes or T cells, whatever. I guess one I don't have here, you know, just the simple standardization across annotations for data sets at different levels of granularity. So I want to be able to say T cell, or I want to be able to say or want to be able to record a particular type of T cell, and I want to be able to group those annotations together. Anyone got? If I just put the floor open now, what are any other suggestions about what you want to be able to achieve with cell ontologies?
David, I would add querying across species from model organisms to human using the same terminology. Ideally across development as well.
Right, but so that could be in terms of querying for the descendants of a particular type of cell, or querying for the for cells that develop from some known precursor cell.
Yeah, I mean, I think on the one hand, it's the developmental lineage from some definable precursor, and on the other, it's perhaps where they are in that developmental trajectory to make sure that we're using the same description for that whole process.
In that vein, I would say that a formal way of describing the evidence for that lineage as well, whether it's inferred or tested.
Yeah, so inferred from a trajectory versus some kind of more direct evidence, I guess.
It could be inferred from observation as well. You could sit down and watch a C. elegans, I guess, as has been done.
Yeah, I mean, certainly in the model organisms, there's efforts to mix sort of classical fate mapping type work with single cell transcriptomics, but if the focus for this is human, then I guess it's not. But I guess that might be something that could be inferred from the cross species side. Is it worth noting? Okay, anything else that you want to be able to do? So I guess projection across, you know.
David, what if what about just comparing the data between the data sets, comparing the data generated between different labs and different users?
A simple cross-searching, indexing on,
Correct, and maybe I think Ed already brought up this issue that different labs may, because they have different biological focus. Let's say development versus adult. People will have different focus, and they may actually call cells differently, even though they, even though they're talking about the same cells. So maybe function of ontologies, maybe not just to have here's the clear name that we all agree on, but also just having a framework, not just naming an ontology framework, but also maybe some sort of computational or programmatic framework that will allow to unify those ontologies between different labs, right? It doesn't matter how you call the cell in your lab, as long as there is some sort of common shared space where all those ontologies can converge on ID number of 5,000, then it will be fine.
Yeah, so the standard, I guess the standard way with ontologies to work with this is you have a hierarchy of terms. So one group might have a different focus, so annotate with a higher degree of precision than another group, or make assertions with a higher degree of precision. Another group might just use a different terminology for the same thing, and in which case we could collect in synonyms. So, so I guess to facilitate this, we need to be in synonyms.
The other thing that I was thinking about is what we run into with the lung is that the current ontologies do not exactly overlap with the view of the reality when you start looking at single cell data.
Mm-hmm.
So I don't know whether it would go in this scheme, but it's somehow reconciling the single cell data with current ontologies and then somehow integrating those two.
Yeah, I mean, I think the cell ontology that's used is it definitely needs more attention, and I think getting a wider set of groups to have input into how that's structured and getting them to be able to easily add terms or make suggestions will help. It's also intended to be cross-species as well, so it might be, and there's been quite a lot of discussion of adding being more explicit about human-specific, different species-specific cell types in there, especially when we record markers. So we have mechanisms for doing that.
I'm not sure if this is appropriate. This is Becky, but also just keeping ontologies up to date as we learn new things from our single cell data.
Mm-hmm.
It's similar.
So maybe that seems to go under there, maybe. And this reconciling yeah, so I think one thing that actually I've discussed in the past with Ed Leans on this call, what's the role of do we want to use kind of example data to define new terms as we're now defining? To what extent do we want to actually link the definition of new terms that come out of single cell work to data itself? And that could link into programmatic ways to facilitate annotation. So if you have some kind of reference data that could be used to test with your own data set whether a particular cell is present. So the Allen data might actually lend itself to that. Do you want to comment on that, Ed?
Yeah, I mean, I think this is a really interesting time, and this is salient for probably pretty much everyone on the call. Of course, we have this body of pre-existing work, but we have this new suite of molecular technologies that's just completely able to define the cellular makeup of complex tissues. And in the brain, most of this is unknown, actually, especially the human brain. And so it kind of flips this around a little bit from trying to link molecular data to this pre-existing framework to the data is the framework. And you link the pre-existing data to that, because you actually have a quantitative way of defining these things, and you have a quantitative way of defining ontologies, and you have a quantitative way of aligning across species. So at least from my perspective, this has changed the situation pretty substantially. That it's not linking into this pre-existing knowledge. It's basing it on these quantitative terms, and then you can iterate off of that. But others may have a rather different opinion about this.
So I guess the challenge to me there is how you mix, you know, the standard sort of categorical way of defining cell types and ontologies, which works quite nicely because you can bring in lots of different factors. It's not just one type of data generally that it's based on. So you could bring in something about morphology and location and transcriptomics and function, versus the data-driven approach, which at the moment tends to be based on single types of data. But out of that, you tend to get, you know, an ontology usually is represented as a graph or a tree, and you get those kind of branching tree structures of classification that come out of the data, single cell data, sometimes morphological data in the fly has been used for that, and increasingly lots of different types of data. So you've got two different ways of coming up with a hierarchy. The challenge there is how do you try and reconcile the two, and is there a practical way to do it? Or at what point maybe does the ontology stop and the data start?
So David, within the context of a framework, to what extent are we talking about developing a new control vocabulary and subsequently ontologies based on all the things that you just laid out, versus taking the pre-existing cell ontology and using that for a framework?
I think, you know, it makes sense in the first instance to take the existing ontologies and to try to specialize them just in a reasonably loose way based on all the feedback and needs that people have got. But as they start to generate more and more new cell types, then the question becomes, how do we incorporate those? Does that make sense?
Yeah, and so the expectation is that if the original framework is robust, then as you identify new cell types, it should not be that difficult to layer that into a pre-existing ontology.
Mm-hmm.
You know, presuming that, you know, you've got epithelial cells, and then we can mark them off with epithelial cell type A, B, and C, and D by various criteria, etc. So that then leads to, I think, a discussion of developing perhaps a working group that would then look at the current cell ontologies to determine whether or not that framework is going to be really robust enough for what HubMap and HCA want to do. I think that at least our initial look at some of the kidney listings in the cell ontology, we found that there were a lot of things that just either were antiquated or just flat out wrong.
Okay, so then the question then is also how practical it is to fix what's there as well.
Yeah.
Yeah, and so probably there needs to be working groups in different areas here. Just it needs to be split up. Mm-hmm.
This is Jonathan. I said I was going to mostly listen, but this particular issue that Ed raised is really compelling, and I wonder if there's an opportunity here to add to what is typically in reference terminologies and ontologies, which is some notion of evidence for the terms and the links that are made in what is really a graph, which is what all these ontologies are big graphs. And so given the data-driven idea, you might have some reference in them to evidence that suggests that these cells are related in a particular way, or naming them makes sense based on a particular morphology or genomic or expression that they exhibit. And because it's going to be constantly moving and changing, and so the things that don't change are evidence that exists. You can of course remove things and put it there, but it's not quite the same as so many things we've done with just getting a group together and declaring out of their general view what the structure of this is. It really, this data-driven idea suggests that one should link to the evidence of every term and linkage that you're creating. I mean, it's a high standard, but I wonder whether that's a way forward.
So within the ontologies themselves, traditionally that's done just by linking to papers. So, you know.
Right. But we may move too quickly for that is kind of what I was getting at. And it may be a data set that sort of shows some evidence or proof in kind of the way that Ed was talking about, such that you can, or maybe a group that applies some standards to apply some kind of rating system as to the strength of evidence, as opposed to just linking a paper, which then requires the user of the ontology to actually go deeper to choose the strength of evidence on their own. So that was just a thought in terms of whether this interim time period, a strength of evidence concept, can help.
I think that's really interesting. I don't know, Ed or anybody else who's already sort of generating hierarchical clustering data, to what extent do you think with a sufficient body of annotation, it might be possible to try and layer one over the other? So is the structure of the ontology backed up by, so we already have, in the brain, for example, some basic subdivisions, GABAergic neurons, and there's some subdivisions under that that have been added in based on categorical assertions. To what extent could we then leverage the data-driven hierarchies to support those hierarchies?
I just want to interject. So the ontologists, and I'm not an ontologist, but they like to think in terms of relationships. And so there is no one set of relationships that sets an ontology. There can be different types of relationships. And from a biology standpoint, you can imagine that one set of relationships would be who's my mom, right? So a cell lineage type relationship, mother-daughter relationships. And I think as biologists, we can find those extremely useful, at least with the developmental biologists. There are also relationships of who's my nearest neighbor, who do I interact with? And that's another set of relationships. I think right now we're focusing on just identifying who the players are. And in some sense, that goes into thinking about a controlled vocabulary so that everyone is naming the players with the same name. And then subsequently thinking about how do we want to organize or be able to express the relationship of these players relative to each other.
So this is just Jonathan. I wanted to say, I think most of the folks on this call understand that precisely as you stated it and would agree completely. And so as do I. And I was just raising the question of where all those relationships come from and how we can give that some notion of strength, and also the same as to whether two things are in fact the same thing or different things, as you point out, is a very core starting point. And there's strength of evidence of that as well. So it's just a way of getting somewhere is what my thought was. I'd love to hear Ed's thoughts, and I'll step aside.
No, thanks. I agree with everything that's being said here. So I guess let me say a couple things. One is that up to a certain level, I think we're definitely looking at the same thing. So the sort of classes and the relationships based on the transcriptome or the epigenome are very much in agreement with a long body of work on developmental biology and many other phenotypes trying to do systematics here. So there's another level of detail or three that you get with these kinds of data that we don't quite understand yet. But it serves as a framework. And I think that this is part of the whole concept here, that we want to get to the understanding of this sort of biological truth. And this provides, one purpose of an ontology is so that we actually define the entities and we define quantitatively what we mean by the entities, and we provide a way for people to link their data to that so that you can aggregate information around it. In the Brain Initiative, the cell census network that I'm part of, we kind of took an interesting approach that is finally going to come to a series of publications soon, which was to kind of take the ENCODE model. and we took 1% of the brain, and everyone threw their methods at it, and then we've been trying to see whether they agree and how well they agree. And this target was part of the cortex. It was part of the neocortex, primary motor cortex, which is a pretty well-defined structure. And we threw various kinds of single-cell transcriptomics, single-cell epigenomics, spatial mapping, patch-seq to look at the physiology and the anatomy of these cells, all sorts of different ways. And the outcome of this is that the transcriptional framework, right now, those tools are most advanced and they give the highest granularity. However, all the omics methods are generally very concordant. So they give you, if you're looking at the chromatin state or you're looking at the transcriptome, they sometimes have different levels of granularity, but there are no major disagreements there at all. When you go to other cellular phenotypes, sometimes there are more significant disagreements, and you get into the real biology of graded changes and things like this, which are kind of a problem for defining concrete entities. But again, the hierarchy sort of helps you in this. Up to a certain level, you can pretty much say that you're getting the same result from however you look at it. And then there's another level of granularity. So I view the kind of what we're doing here is we have a chance to sort of quantitatively define biology. And at the moment, this transcriptional readout gives you many positive things. It gives you a way to quantitatively define them. It gives you the molecular code for 1,000 hypotheses about the genes that are responsible for the cells. And it gives you a metric for measuring similarities and relationships across things, which turn out to very strongly mirror developmental biology, as it turns out. Maybe not a terribly big surprise. And so I guess I'd like to ask people whether they feel that I sort of view that we're kind of in a place where we could follow the model of the genome, and the ontology is a way of kind of formalizing that. But it's time to define the entities and to name them, and to really build the system that's then testable and can be iterated. If you remember, the first versions of the human genome had like twice as many genes as the next version, so it underwent rapid change, but that didn't stop it from being like really transformative at the beginning. So that's kind of how I would see that. Rather than saying let's try to piggyback this on a very qualitative, and at least in the brain, the cell ontologies that are available now are highly incomplete, like really incomplete. We have data sets coming now that cover the entire brain and serve as a framework to then link other kinds of information to it. And I think we should capitalize on that personally, and that's also the kind of common language across all of the organs that we have here. And that's part of the purpose of bringing all these groups together, is that we're all using the same metrics.
Would you say that the very high level classifications are useful, but as soon as it gets down into details, it's, sorry.
I mean, I think it's all useful. Some of it is there's just more information and there's more obvious agreement at the outset. But that level is deeper than is currently in the ontologies.
Mm-hmm. Yeah, I mean, partly I think it's the history of the cell ontology hasn't had a lot of fun. We've got several thousand cell types in the Drosophila brain that are quite well characterized, and we're linking that all up to data already, but we've had funding to do that.
I'm just going to say that it probably depends on your tools as a user, too. If you use single cell data to define many subcell types, but in your lab you have 4 or 5 antibodies, you can only go so deep. So the hierarchy is very helpful in that way. My other question, a question I had was, when we speak of testing, I'm thinking of testing proposed ontologies or new ontological terms. As ontologists, are you thinking about how to test your given ontology, and how is that done? Is it done cross species or with just different data sets to see how it sorts out?
Yeah, I mean, I guess they're not generally empirically tested. Well, it depends. I mean, in the case of, long-standing projects that have been quite closely linked to data, then they're very much shaped by the data that we get. So, in Drosophila we have several hundred, soon to be several thousand cell types with morphologies attached and connectomic data attached. So we don't have that for the cell ontology. There is more kind of semi-empirical work. Well, there's in the case of an ontology like Uberon, which has been used to define, which has been used in lots of different projects for annotation, but also to structure other ontologies by anatomy. Then there's lots of feedback that comes from these other ontologies, such as the gene ontology, phenotype ontologies, disease ontologies, which has helped shape and improve that ontology. But I think there hasn't been as much effort on the cell ontology, but maybe this is an opportunity to kind of start feeding in data into structuring it better. I hope that
I'm a little worried that this touches on a lack of funding attention to the general world of ontologies, which has been, I think, unfair in the past.
Yeah, I think that's fair to say.
So I really think that one of the messages to make sure we bring out of this too is that everyone acknowledges the great need. There needs to be more money ponied up for that need.
Yeah, and I think there's been especially lacking in the case of the cell ontology. I think things might be starting to change with some sources of money coming in. And I think we need, I think it's very important for any of these ontology projects that they're very much tied to applications. If you set people off to build something, but it's not clearly tied to an application, this is why I put the use cases up the top here. If it's not clearly tied to an application and a set of use cases, then people will build something kind of plausible, but maybe not completely useful.
In regard to this, Jonathan, if I jump in, this even where they are tied directly to use cases, I think we've had trouble funding the parts of that work that makes everything aligned with the reference terminologies. So it's both the development and the use, I think, has really been underfunded.
Yeah, and I think the good opportunity now is there needs to be a mix of some core funding to do the basic core work to support releases and there's just the general quality control and all of that. But I think it's a good opportunity to push out to the community and get lots of community input and the ability of the community to maybe edit and suggest and quickly turn around terms. So I think that's the only thing that's really going to work in this situation with everything moving so quickly as well. So I'd see this meeting as an opportunity to try and pull together people who are interested to contribute. So for the cell ontology has just started up regular meetings again, and we're organizing some training sessions as well. So I think that's getting the word out for that and getting some online training sessions, I think, especially in this time when lots of people are not in their labs. Maybe we can actually, people might have a little more time to be involved in this kind of work.
It's an interesting point about clear messages that should come from our groups the NIH might hear. I mean, for example, a message could be, we see the need for a cell ontology like the same kind of scale as gene ontology to map the work of all of these different efforts.
Yeah, and I should say that the cell ontology really has kind of branched out of the gene ontology in the first place, but lots of the structure was actually derived from that's one of the biggest users. Okay, I think we've hit a little more than the 45 minutes for the initial session. I haven't actually had dinner yet, so is everyone happy to break for 15 and come back into session 2, and we'll try and get some action item points? Should we try to make a start? So first of all, I just wanted to open it up and say, to see if anybody else has anything they'd like to contribute that's I'm conscious it's been a relatively small group of us doing most of the talking. Are there any kind of key points we've missed, or anything anybody wants to share about their use of cell ontologies so far in the work that they do, and frustrations and how it might be improved? So I guess we've got kidney and lung people here. Presumably have had some experience in using or trying to annotate cell types.
I know I'm one of the people who's already talked, but I'll just talk anyway. And I think that one thing that in collaboration with the KPMP, the KPMP has tried to bring a bunch of us together on the kidney side to try to work on what we need from an anatomical ontology. And in bringing ontologists together with us in a meeting we had in January, one thing that has always been clear is it's difficult for the domain experts to understand ontological frameworks, speaking for myself. And there's this gap between the two. And bridging that so that the people generating the data can contribute more readily to the ontological work that's difficult to do without the contributions of the data generators and vice versa. I think that's been a concern for us and something we've been trying to work on together. Do you find that to be the case?
Yeah,
Often,
I think it often is. I think, you know, a closer relationship with the data generators and these projects definitely helps. And I think in some cases, sort of, the ontologists have been brought in a bit later to try to work with people rather than, but, you know, if we can work with people from the start, that helps. For some of the fly work I've been doing with, for the Fly Cell Atlas project, we've been getting some community review and feedback. So we've been developing various ways to share kind of spreadsheets that summarize what cell types are available for particular tissues, for example, to get feedback and what references we have on those. And there's definitely, you know, in areas that haven't had any attention for a few years, things tend to get kind of out of date. So definitely building a closer relationship. I think on the anatomical side, I feel, you know, atlases need controlled annotation, and ontology as controlled annotation needs atlases for reference. So if we can, on the rather than the cell ontology side maybe, but also certainly on the anatomy side, if we can hook the ontologies into reference atlases, which we've already started to do, you know, we've got mappings from Uberon into the Allen Brain Atlas standard reference atlases. I think that will help a lot, because then there's, you know, not just a textual definition and a reference or some kind of graph structure, there's a link to an image, you know.
I worry that one thing that may help with closer collaboration, aside from beer and pizza, could actually be if the data generating groups understand clearly sometimes, often it's fine, but understand clearly that there's a benefit to them, like there's a return on that investment of time. They feel like it's extra work, but it's actually not. It's part of the foundational work that needs to happen. Yeah,
and I think that it's a good time to push this right now, because when people are working in small groups on their own and there's limited data sharing, then the you know, the payback from standardizing your annotation is relatively low. But now we're moving into an era where everybody's sharing a lot more, and they want access to other people's data and to be able to understand it quickly. Then the payback is much higher. So
so that's what we heard this morning, right? Was that if cells were annotated well, and the tools that some of you on this call are generating, I think, if they're broadly applicable, that accelerates that payoff. It makes it very direct and speeds everything up.
Yeah, I think so. And I think you know, there's often frustration with turnaround with getting stuff into the ontologies, but that there at least needs to be some kind of core level of funding so that we have the people to do the basic fast turnaround and to incorporate feedback. It doesn't require much. You know, you had a couple of FTE on this, you could do a lot.
So what's the phrase? Work for free or work for a lot, but not for cheap, okay?
And I think
You know,
if you're making, I don't know, I think it's not particularly clear, I would say, to the community, like the broad community, how would they go about like improving an ontology or providing feedback. So I think people might just go and have a look at the ontologies and think, oh, that's wrong, or I'm not going to use that because it doesn't have what I need, and go somewhere else. Whereas if there was a bit more ability to provide feedback, or an easy way, if you make it easy, then people are probably going to be happy to try to improve things. But if it's a bit of a burden to try to figure it out, they're less likely.
Yeah, and I think maybe one thing that could help a lot is to have some level of outreach as well, where we do a certain amount of training so people are clearer about the advantages of using the ontologies and what's there and what they can use and how they can best give feedback. And, you know, potentially training so that people can, we actually have had a certain amount of community edits recently, people getting in contact, and we've done very quick training. You know, people who are technically minded enough to be able to do a GitHub pull request. We've had a few edits come in like that recently from single cell, you know, based on new cell types found from single cell data. So we're more than happy to train up the community as well, but I think it will still require enough funding that there's somebody there to make releases and do the sort of basic work to keep things ticking over.
David, one way to increase the community usage may be to align the ontology very well with some of the tools for people to be able to map their data to. So what I mean by that is, you know, as these transcriptome-based classifications or reference classifications kind of get finished, then the next kind of thing is to develop tools like BLAST, but for your cell types, for your cell type data, so that if you generate a new data set, the first thing you do is you align it to the reference and you transfer the labels onto your data set.
Yeah, so there are some existing tools like that. What's it called? SC Map tool developed at the Sanger by Vladimir Kiselev and I'm trying to remember the others. Anyway, that's so far takes you know, just textual annotations, but there's no. It's easy enough to find a way to get the ontology identifiers in there as well. I've done a little bit of work myself on extending Loom files to incorporate mappings into cell ontology terms. So I think, or into any ontology terms. I think you know, getting these getting the tools out there to the people who are doing analysis as well in a form that they can use, I think will help.
So from the lung working group, our experience here is that people will start thinking about ontologies and start using them on a need basis, basically. So there's multiple data sets. You want to compare data, and then you're just talking a different language because they call a cell different than you do, and you know somehow need to agree on the middle ground. And that's where the ontologies come in. When I mean, a lot of the people doing this sort of work at this moment don't have a background in ontologies, are not per se interested in them, but they want to use them. It's a tool for them to progress their studies and to be able to integrate data sets and to be able to answer additional questions because you need more data, more donors, more whatever to be able to answer that. So coming from that perspective, if we want the ontologies to be useful, one of the things, there's a lot to gain, I think, by outreach and So for us, talking to, within the lung community, not talking to the single cell community per se, or the ontology community per se, but talking to all these people doing clinical studies where they also do some single cell sequencing and say, look, here's a tool. If you run your data through this tool, it'll actually return cell types to your data, to your clusters. That is actually in agreement with what everybody else, has, there's a consensus ontology here. So those sort of tools that actually help people progress their research project, I think, are going to make a huge impact. And then people will also appreciate the value of it. And so it's really from a use case basis, I think, where this, this will help develop the ontologies as well.
I think this is a really good point, and I would say that if it's understood and made clear that there's a certain level of the hierarchy that tool is applied to. Some pushback we've had GoodMap from some of the data generators was that, well, if you give us your Seurat object with your clusters named, we can share that with people, and they worry that it's their interpretation of their data that's labeling those clusters. They want people to be free to do whatever they want. There is the fear that the tyranny of the default, if you will take over, and people will just accept that without critically thinking about it. Those who, go ahead.
So what we've been thinking about, and what we've actually organized, but that got canceled through the whole coronavirus issue, is basically have a meeting, and I think the kidney people are a few months ahead of us here. Bring in the experts on cell types and the domain experts, bring in ontology experts, bring in the single cell people and the computational folks, and then sit together for a few days and really discuss it out, come to a consensus that people are also, people who are not going to go too deep into this are going to be happy to use that. And then, of course, in a consensus document, you can clearly say, these are areas where more interpretation, more data is needed, and, it needs development as we go on. Be careful, this, this annotation that we provide is not cast into stone, but it's just something, a starting point for you to help you on your way. So I think people are critical enough usually of their own data to be happy to still be critical and to reflect on the value of whatever they find.
So would it be fair to say that that having a better way of recording evidence and provenance, so it's clear whenever you pull back an annotation what that's based on, it's based on the particular set of markers it's based on, for example, would help? And should we be discussing standards, metadata standards for the kind of standard data sharing for cell type annotation, at least in the standard files that the community uses to share single cell data? So I know there are extensions to Loom files, for example, that allow clustering to be embedded and then clustering data to be annotated. There's also the HCA DCP have been coming up with standards or provisional standards, at least, for annotating cell types in Loom files. Is it worth having some kind of working group that solidifies a standard like that a bit better, so that there's a standard way to record evidence, for example, marker sets? There's some work on that in the Fly Cell Atlas community already. Any comments on
that? So there's two ways to go about this. One is where you have a data set and you use the clustering and then the markers that come with that as evidence for your cell type annotation. And then you start integrating data sets at a level of annotated cell types or clusters, if you wish. The other way to go around this is to try and integrate the data as more or less raw data and then cluster the whole thing together, which has a whole bunch of other problems. But once you're able to do this, you actually increase the power to also start detecting like rare cell types and transitional cell states, etc. You know, all the middle ground there, which is another way to approach it. And then in that latter scenario, which I think what most people have in mind for things like the DCP, I'm not so sure how much of this evidence from the original clustering is going to be relevant, because the clustering is always based on the background of that data set, right? So as soon as you add more data, the clustering will also change.
Yeah, but at least if an annotation was based on a particular clustering and you had a way of recording that.
Yeah, no, I can see that makes sense. Yeah. So yeah. I mean, I'm not an expert in this, but yeah, if that would be possible, and you know, the original cluster annotation and the rationale for that would be retained somehow in an object as metadata somewhere, that I think would be useful.
I think it's a tricky thing to come up, you know, to come up today with the ideal solution for this, because the tools for mapping are still pretty nascent. You know, there could be huge systematic biases in the data generated by any particular site. So the joint clustering is appealing, but has some challenges with it as well. You know, a lot of these original references are at least being generated by like single sites doing very, very large scale systematic, highly QC'd things. But I think as long as you just track what you've actually done, there can be multiple ways of doing it.
I know within the Kidney Precision Medicine Project, you know, so we've kind of got a couple phases here. In our first phase, we are working directly with ontologists in Ubron and the cell ontology, which are the ones we decided to use for anatomy and cell types, respectively, to shore up what we think are, you know, base terms, or, you know, concepts that should be in those ontologies, those reference ontologies. But then we're also developing our own ontology that's kind of writing on top of that for things where it may not be ready for prime time, right? We're seeing some clusters say this, but it hasn't been, it's not canon yet. And so we're developing a, you know, specific ontology for those terms and others that are specific to our program. And then as, you know, just over time, those might get promoted up to, you know, the actual reference ontology, or maybe they'll go the way of the dinosaur if we find out that that wasn't, you know, that data didn't hold up. So I guess it's important. It was an important thing, I think, for our consortium to recognize that we didn't have to nail every term at the outset, right? Like that there's levels of detail that we can, you know, that we don't have to go to in particular in the reference ontology if that was debilitating for us. I don't know if that makes sense. Sounds like it doesn't. That's okay.
I guess what we're arguing for is a system that's flexible enough to be able to allow these different applications.
Yeah, and I think the people get a bit uptight sometimes about adding stuff to ontologies, and that they worry that, you know, it has to be, that you have to have some high degree of certainty. Though I see it more as an operational thing, you know. Is it sustainable? Is what we're adding a good enough standard for now based on the data that we've got? We don't want to, if there's something you're really uncertain about and you think, you know, might go away, or will change, the community will change their minds about it, and in the space of a year, it probably shouldn't go in. But if it's something that could be stable, reference to data, will work for 3 or 4 years and then be revised, then that's fine. So I think there's kind of a balance there. Sometimes things get kind of paralyzed because nobody wants to stick their neck out and define a bunch of cell types, you know. But at some point, I think it's helpful to do so, even if, just like drawing lines on an atlas, you know, there may be a future release of an atlas, or a future, you know, releasing an annotated genome reconstruction. Those things change, they just need to be stable enough.
Yeah, so the other thing here is that a lot of the applications of what we're doing is going to be in the context of disease. And what I see, at least in the lung, and you know, I'm as guilty as the next guy, whenever you do a disease study, you invent your own cell types or cell states that are disease associated because it sells a paper and it sells your next grant. However, I mean, so I would argue that having an ontology as a flexible thing is actually a good, a good thing rather than a static cast in stone, because then people are not gonna use it very effectively. And being able to debate and we make this observation. Is this really a new cell, like a transitional cell state that is specific for disease, and it's stuck there? And compared to the reference ontology, how novel is it, and how much can we learn for therapy based on this? Those are the sort of papers people will be really interested in, I think. Not so much the basic, like the hardcore biology of it. What is a cell type? Where does it end and where does it stop? Or where does it start and where does it stop? So I would argue as flexible as possible. Degrees of certainty would certainly work for me. This might be not the best audience to say this to, but I'm happy to hear your opinion about this.
It seems to me that.
Yeah, go ahead, Ed, please.
I guess I just wanted to say, it seems to me that this has already been tackled in the genome space, where there's lots of revision that goes on as more evidence mounts, but the model hasn't been to only put out the things that we're hyper confident about. And I think there's a danger in not naming things that we think that we see at some point in time, because if we don't do that, they effectively don't exist. If you put them in and then no one can find anything to corroborate it over time, then you collapse it or you get rid of it. Also, people calling things different names has been handled very well with aliases of gene symbols before. I don't think these things are problems for an ontology.
My question had to do more, also following on that, is do disease cell types belong in the reference ontology?
Actually, that's a place where I think you need sort of parallel structures or some kind of phenotype ontology for that, because I think I see the purpose of things like the cell ontologies or anatomy ontologies as being kind of canonical references, and everything depends. Biology, if it does anything, it varies. And you can't take into account every variation and try and stay sane when building one of these things. So it's okay. Wild type, canonical, these things are semi-mythical maybe, but they're certainly very useful part of biology. And I think and I think it's also worth, the bread and butter of working scientists is the fine detail and the controversial stuff. But there's a hell of a lot of stuff that's been agreed for, the past 100 years and is not likely to change. And that stuff's really useful to get right in an ontology. So, some basic subdivisions of lymphocyte cell types, basic subdivisions of brain cell types, basic bits of anatomy. They're agreed and non-controversial, but getting that stuff right is really important to make these things usable. And in that sense, canonical reference makes sense, I think. Sorry, go ahead.
I was just say the analogy to that Ed was making with the genome. You don't put disease genes in the reference genome. There is another structure for that. So you're either a healthy Craig Venter or you're not, I guess, is what it comes down. But that needs to be in place, I think, or at least a process that we could grab hold of to point out to people where we've discovered a new cell phenotype that's related to a disease.
Yeah, at the same time, if you take the parallel to genomics, you would try to capture the variation in there. So all the SNPs, even when the SNPs are disease associated, you'll still annotate that variation. So the same holds true for and this is of course becomes semantics at some point, but the variation of normal, how to what extent do you capture that?
Yeah, I mean, there ends up being a lot of gray areas with this stuff, just as there is with any phenotype where, people are recording subtle trait associations in GWAS, for example, which may fit within what you could consider canonical or normal. But,
could you envision a way of sort of having a track, like a SNP track, against an ontology? Or that's a type of tool you might envision for the future, a graphical tool of some sort?
It's an interesting analogy. I mean, we already have phenotype ontologies that reference canonical ontologies. So you can take Uberon and you can pull a set of phenotype terms associated with anatomy. We've got less in the way of cell phenotypes right now, but it can all be built using the same structures. And there's been a reasonable amount of money that's gone into standardizing phenotypes over the past few years. I'm quite closely involved with that. So yeah, I mean, linking it into the phenotype work as we do it makes a lot of sense. It just needs to live in a kind of parallel but linked structure. So we could have that as one of our aims as well here. Yeah, whether the kind of linear, unfortunately, nothing we deal with is one-dimensional like sequence. Okay, is there anything else? How are we doing for time here? We've got half an hour, I think. It's meant to be finished by 9 my time.
Yeah, we may want to look at the blue box section or whatever of this to see what we should be talking about.
That's exactly what I was thinking. Okay, a title to your collaboration. Oh, I think you can have more than one blue box if you want. I think that was one of the things they told us. So it makes sense to say the main problem we think we can solve by collaborating is does this make sense? Collaboratively building it, so with building a cell ontology that's fit for purpose for annotating and querying across the across single cell transcripts. Sorry, go ahead.
Hi, it's Alexander Misharin. Just to jump on this, there is an ongoing effort, and a few people actually mentioned this. Maybe it's worth specifically pinpointing that Piotr Harchenko at Harvard is leading this effort. I think that would be just a direct point of contact because people mentioned that a couple of TEs can get this work done, and my understanding is that Peter finally has secured funds to do this work, and there is a GitHub repo where this work is at least outlined. So maybe that would be direct actionable point to actually.
Yeah, I think so. Peter's work is more, as I understand it, working with the community to make an annotation platform that can consume cell ontologies, but also brings in annotations and the needs of the community more directly. So yeah, I think it definitely has to be something that would work with what Peter's doing.
I've put a link to that in the chat, and this seems like a good point to call out that Peter will be co-leading a session on annotation tomorrow in the second session. So interested members may want to join that session.
But I think you're right, though, that his intention is not to build an ontology, is to aggregate community annotations and possibly even to apply an ontology to data sets that people put in.
Yeah, so I think it's kind of complementary. It pulls in lots and lots of use cases, and there's potential to use the platform to reconcile different use cases and feed that into an ontology. Okay, should we put a reference to that? Here's the chat. Oh, this is different.
The model.markdown file is the most interesting part to me.
Oh, okay. Right, so there's a slot for the cell ontology terms in here. And there's confidence, which is nice. Okay, I mean, it definitely, it's past time to be discussing. And I think that, you know, there's clearly a need for sort of rapid work on being able to add new terms and maybe converge on consensus, but also at some point, I think it needs to be solidified into something a bit more concrete that people, you know, I've seen lots of things that I've seen community annotation efforts before that have been partially successful, but also ended up with a lot of annotation, which further down the line, database curators have torn their hair out trying to use. So I think there's a balance to be struck. Okay. This one. So actually, can we mine some of the things out from what we wrote earlier on this? I think there was a few things about working groups. Oops. Oh, what did I press? Sorry, I think I managed to close it. So I suggest working group to audit current ontologies for coverage in particular domain areas. There's one. So reasonable to go in there. Is that, maybe that's, and there was a bunch of stuff on outreach as well. Maybe those two things are connected. Would it be our working group? It's going to be different. It's really, maybe it's more a matter of reaching out to particular communities to get an audit.
Yeah, definitely. We, in the kidney space, we found that, you know, we needed particular kidney domain experts in the room with ontologists. So I would feel like a single working group would probably get too mired in differences between the different domains.
Yeah, I think so.
Mm-hmm.
And that would be for each of the different, does the hub map structure work there? And is there enough collaboration across hub map and the different HCA groups for that to make sense? Maybe that's not for us to decide at this point. This should be facilitated by outreach.
I think it works best within organ systems.
Yeah.
There are also some groups who are working on multi-organ systems at lower resolutions, Guojiwo's comes to mind as a topical example. That could provide some utility in looking at stromal cell types and other types that span across tissues.
I think a lot of this will involve like grassroots activities of the different sort of organ systems where people have a need and then they'll just start, you know, rather than reinvent the wheel, look around. So if there's like a blueprint how to come from people that realize they need ontologies to actually implementing them or developing them or applying them and contributing to the core knowledge of it, that would be great. I mean, we've been basically racking our brains within learn how to do this properly, and then, you know, we started looking at other organ systems, and some of them are more well developed, like brain. So we just asked, like, how do you do this? So if you know, if we could feed that to the community at large, I think that's going to be extremely helpful, and a lot of people will actually get, you know, interest in this.
Definitely echo that.
Yeah, at the same time, you know, I think it's for each domain there will be the most engagement and the most knowledge from its internal user base, but at the same time, you don't want to set up a system that is catered for one that doesn't apply to another very well. And so for, an example of this is in the adult brain, almost everything is post-mitotic and has been for 50 plus years. And so that community is all about clustering and calling those things cell types and not about developmental trajectories. And you go anywhere else and things are more about trajectories. And so I think some cross-fertilization is really, really important as well.
So, yeah.
David, I apologize if this was covered. Is there, I guess, a contributing guideline for the cell ontology? Like if I wanted to go and let's say I was really interested in the brain and I thought that there were some additional cell types that weren't covered, is there a way for me to fork the cell ontology, go do a bunch of stuff and then PR it back?
Yeah, there's been quite a lot of sort of cleanup and changes in the repository structure recently. We have trained up a couple of people we've had a couple of tickets recently where we've actually just spent 20 minutes, half an hour with somebody to show them how to do pull requests and how to do the edits, and we've had pull requests come in. So it's doable. I think we need better documentation because we've actually changed. We have a standard ontology development kit platform that's built on GitHub that the cell ontology now follows, and lots of other ontologies follow that give us sort of Travis integration and unit tests and stuff, integration tests. So because of that, a few things have changed in the way that it works. But yeah, it's doable. It just needs a little bit more doc.
Yep, it's Chris Mungle here. I'd say,
hi, Chris.
Hey, just the same kind of rules that would apply as to any kind of software project. You know, if someone comes in, doesn't do any upfront coordination, and makes a giant pull request with thousands of changes, you know, the likelihood is that might just get closed, and we might say, hey, we'd rather you made this kind of like smaller chunks, or maybe let's coordinate in advance. So it's really the intent is no different than we run things just the same way we'd run a kind of like software project, and we'd want as many kind of community contributions as possible, but we'd want to manage how that gets done.
Could you give me a sense of how many such contributions you've had and your success rate at, I guess, converting interest into community contributions to the ontology?
I would say there hasn't been a huge amount recently. There's been, you know, more on other ontology efforts that, but like I say, there hasn't been as much. I think there isn't really core funding for the cell ontology, so there hasn't been a lot of manpower to put into this. That's starting to change. There's some money that's coming into it now, but I think we still need a bit more to really be at the point where we've got good core funding to support this. But it doesn't take much, as I say. I think, you know, another FTE on it or 2 FTE on it would go an enormous way to making this all much easier.
Got it. Thanks for the explanation.
Yeah, and I think I'd really, as well as better documentation, I think we need, part of the outreach should be, well, actually, I'm already planning to run one training session later in April with some people from BioHub on editing the cell ontology. So we're starting those up, and I think I'd like to do a lot more outreach to train the community to be able to do those pull requests. Was there anything else? I think there was some interesting stuff on provenance that we haven't covered here. Sorry, where was I? Standards are required for recording provenance of annotation. I mean, I guess that's something that's probably going to be covered tomorrow, but we've got multiple channels going on here. I'm flagging a bit here. Anything else on the for recording provenance? Right, that's something that's a need, but what are we actually gonna? What do we want to do? Okay, so.
Does everyone here agree on what an ontology needs to contain? I just asked the question because I think we don't necessarily need to have the same form, but if we had some kind of agreement on what the content should be, then an interchange format would be a reasonable target.
I mean, we have some pretty standard interchange formats for ontologies already. We have formal languages and.
But does everyone agree on those?
That
was sort of the root question.
Okay, so what should be? I mean, format-wise, I think yes. What should be in an ontology in terms of the biological assertions is I doubt there is agreement here on that, but that's something that's hard. Chris, would you like to talk to that at all?
So sorry, what was the question there?
I guess there was two questions. Is there a general agreement on what a reasonable interchange format could be for an ontology? And then at a deeper level, do we agree on the types of information that should be contained?
Yeah, so the interchange format is OWL. And so that is definitely the standard interchange format. It's not necessarily the format that you always want bioinformaticians working with. So there's also an alternate kind of like JSON serialization that abstracts things in a way that are kind of easier for people to work with. But OWL is the main interchange format. In terms of what the content should be, I think there's broad agreement, you know, and this is part of the reason for the existence of the OBO project, is really to kind of like just standardize, you know, at a very basic level, some of the things that that an ontology should have. So at the very minimum, you know, just standard things like each ontology class should have an identifier that doesn't embed semantics in the identifier, but it should also have a primary human readable label. It should also have aliases, you know, just like, you know, gene symbols have aliases and so on. And you can even annotate those aliases to say, you know, this alias came from, you know, this publication, or this is an exact alias, or it's an inexact alias, all that kind of thing, textual definitions. So, so there's broad agreement on the kind of like the core, or at least recommendations on what some of the core things that it should include. And then depending on the domain, you know, for something like a cell type ontology, you know, there's broad agreement that there should be part of links going up to the tissues that those cells are part of, and develops from links, you know, expressing the lineage. So, yeah, but within that framework, there's still room for kind of like, you know, customization depending on particular use cases. So does that sort of address the question?
It was informative. Yes, thank you.
Yeah, it's a big question, and I'm kind of new to this space, but I'm wondering what other kinds of things would you put in the besides this, you know, part of and whatever whole part relationships. What kind of ontology support is there for inferencing or classification, and how much do we? I know there's lots of support in various languages, but how much of it is typically used in this domain?
Maybe Chris, do you want to go?
Yeah, I mean, there's definitely lots of support for, I mean, inference is one of those kind of loaded terms. So we do a lot of inference within ontologies, but it's more a kind of like deductive kind of like inference based on very kind of like crisp, precise definitions. And some of that is maybe kind of like trivial, like, you know, a cerebellar interneuron is any interneuron that is part of the cerebellar region. And then there's support for kind of like using the part of links within the tissue ontologies to kind of like say, okay, if it's this region that's in the cerebellum, then it's in a cerebellar interneuron. What we don't have support for within ontologies, and this is, you know, a feature, this is by design, is maybe the kind of statistical inferences that you might want to do in a data-driven way. So that might be something that, you know, happens outside of the standard ontology inferencing framework, but we'd want some kind of like way of kind of like connecting these more statistical definitions to the kind of the crisper logical definitions that we typically have in ontologies. And that's, I mean, that's kind of like, you know, the big question really in a way when it comes to like these kind of more ontological top-down classification approaches versus the more bottom-up data-driven approaches. And it's still kind of a bit of a research question, I think, how best to align these approaches.
Thanks, Matt.
Do you want to add to that at all, David?
Yeah, I mean, so I think we've got, you know, a reasonably rich and growing set of types of relationships that we can record. You know, for example, various different types of relationships between parts of neurons and parts of the brain. So where synapses of different types are, for example. But and then some ability to do a certain amount of spatial reasoning with that as well, certain amount of temporal reasoning that could be useful for the sort of crisp categorical reasoning, but that could be useful for modeling development. But I think the interesting thing now is where we're actually, you know, those sort of general assertions are encountering real data, and we're seeing that in a big way in the fly world with complete connectomes. So I think there's work that's needed to try to reconcile those two, and that's quite urgent. But the inference, there's a huge amount of inference that goes on, but mostly it goes on as part of construction of the ontologies. So that crisp deductive inference, it goes on in ontology construction and makes it sustainable. There's fewer projects actually use it directly. Mostly it's outputting a graph and then people use the graph to group things. So we've got about 10 minutes left. Do we need to tighten up? We didn't give this a title. I want a title. And then do we need to tighten up the recommendations here? How are we going to do the outreach to start up these working groups? Is there a concrete, are there some more concrete action items here? I mean, there's a certain amount that we can do within the cell ontology right now. I guess one of these, open up the cell ontology meetings to and widely publicize them might be a good starting point. Oops.
And just in the spirit of, you know, use cases and things, it, you know, it'd be, if you're going to do that, it's still to draw in, if you're hoping to draw in domain experts, you know, harkening back to what we talked about a long time ago, is showing them the return on investment that they would get from participating in something like that. You know, really target it. If you're going to do some outreach, target it to that audience.
Yeah, I mean, I think that could be, so one thing potentially we could do there, I mean, so I recently worked on some stuff for the HCA where we did a small amount of quite targeted work to get a bunch of cell types in for data that was urgently being ingested. I think we could potentially choose small focus sets, or get them to suggest small focus sets that we could do some detailed work on and show, you know, just as little pilot projects with the resources we have to show what we can do.
Absolutely. And obviously with the HCA, you've got subsets of domain experts already that, you know, that you can pick from.
Did we address the question about interest in organ-specific groups for the cell ontology?
Yeah, I guess that's the working group to audit that we actually need. So the aim maybe should be for these. That makes sense. Review and contribute. I mean, I think in the initial instance, it's just to review. Well, I guess any review should give feedback that's actionable. So. And then I guess we haven't, do we want to leave out the thorny question of how we start trying to incorporate data into cell type definitions? I guess that's still in the discursive phase. There could be some potential for money to work on that in the near future, but
yeah, I guess the question is whether we're trying to think of something that's tangible we could start a working group on now versus aspirational, and what the field sort of needs at this point in time that's not there. I would advocate putting it there as an unmet need.
It's worth having a working group to discuss, or anybody too early to commit to that? Or
It doesn't seem too early to me.
okay, potential working group. And then I guess there's probably a broader folding this standards for cell type annotation for recording provenance and evidence, that probably needs to be folded into some broader working standards working group. Um, does that all seem reasonable? Shall I just review these and people can yell if there's issues? Recommendations, core funding's needed. Guess we need to get people to apply. We need working groups to audit current ontologies. I guess we're talking about the cell ontology here, really. Set up organ-specific groups to review and contribute to the cell ontology. Reach out to existing annotation standards efforts. Outreach and better doc needs to train the community direct by pull requests to the cell ontology. The standards are needed, but maybe can be folded into other working groups. And I guess there's a whole session on cell type annotation tomorrow. I guess that can cover it.
The cell annotation is not going to happen tomorrow because a lot of it was covered with what you guys were doing. So, so it's you guys are it.
Oh, okay. I thought that would be Peter. Huh.
Oh, did that just happen?
When did that? That's kind of disappointing.
So the subtopic on cell annotation is basically you're covering what it was going to cover. So what we have left in the cell type annotation is hang on. Is expert annotation tools and initiatives, automated cell type annotation, and integration annotation across data modalities. Those are the three topics which will be under the cell type annotation.
Okay. So might be a bit of a stretch to fit that in there. This is more. So this is more the not the ontology side, but the standards for recording the annotation, which we haven't really covered too much here, and I think are really important. So,
David, I wonder if, in the spirit of this meeting of bringing together these major consortia that have different focuses, but they all have some of these common needs, whether we might propose a different model for funding, which would be something like each organ-centric funding group contributes some resources to a standard, and then it gets applied to their organ.
That makes a lot of sense to me. I mean, I've certainly, there's been ongoing discussions, for example, with the Wellcome Trust open science initiatives, whether a better way to fund a lot of the standardization efforts would be to provide smaller amounts of money to large data providers to work with the so this has come up in the case of some of the flybrain work, but I sat on the panel for a working group at the trust a couple of years ago on open science, and there was quite a lot of discussion of this there, alternative funding models. So
this is Jonathan. The Common Fund programs are doing a similar thing. So there's 13 Common Fund programs, data coordinating centers, and there's an RFA specifically that only those centers can apply to do inter-consortium activities. So they're not specifically focused on this topic, but that approach of taking a large center and asking it to do specific things with another large center does seem to be coming along.
Yeah, and I think so, Chris. I think some of the kidney people have reached out to you already on this. I think the difficulty has been that the turnaround's been very tight, and it's been hard for us to get people in place quickly enough to be responsive.
Agreed.
Yeah, correct.
Yeah, so if we could do that, but not have such a tight turnaround, and we can actually get people in place to do the work, I think we could probably do it more efficiently.